Thank you, Brad. Thank you for giving me this opportunity to present what I think is my most exciting research project to date and most promising. Um, a research project in which we discovered a new small molecule that could be used to treat and hopefully cure type 1 diabetes. And what I mean by small molecule is a drug or a medication. In this case, that could be taken orally as a pill, as opposed, for example, to a surgical procedure. So in this case, a small molecule means a drug or medication. And I would like to start by the end by thanking everybody who was involved in this study. Is a very outdated picture of my research group. As you can see, we do not respect social distancing guidelines. So obviously some members are missing, but the work I will present today is mostly the work of Nidish and Wayne right here. And before I go into the topic, I'd like to say a few words about myself and my research. So obviously I was born in Montreal, hence the huge French-Canadian accent. I trained in Montreal and then in New York. And for almost 25 years now, so it's going to be exactly 25 years this spring. So for 25 years, I've been studying pancreatic beta cells. So I had this huge passion for diabetes research, even as a kid. And I've always been interested in pancreatic beta cells. And what we do in the lab, so we try to identify new genes that regulate beta cell mass. With the goal being to, so the reasoning is that if we identify those genes that govern beta cell health, then we could harness them pharmacologically to regulate beta cell birth, death, and hopefully life in an attempt to stimulate beta cell regeneration and prevent beta cell death. And we do so using a combination of approaches. So we use cell lines to go for speed and quantity of material, but we also use human islets to make sure that we're as close as possible to the human physiology, and obviously genetically engineered mice to get a whole physiological model. And we try to identify and validate those genes in each one of those models. And today I will tell you a story about one of those most promising genes that we have discovered called LYN, L-Y-N, LYN. So what we discovered is in cells, activation of LYN promotes beta cell survival. So we protect beta cells, and then we also stimulate beta cell proliferation. So we stimulate beta cell mass expansion. And we also, and when we inhibit LYN, we provoke cell death, and then we also prevent beta cell mass expansion. And I just wanted to show you one result out of many, just so that you know what we're dealing with, what we do in the lab. So for example, here in these pictures, we stain beta cells in blue, and then we stain those that are dying, that are in the process of dying in pink. And you see here in the control situation, there's like 5% of the cells dying, but when we inhibit LYN with two different compounds targeting two different portions of the genes, we stimulate or we provoke beta cell death. So that's just to illustrate what we are doing in the lab. So at this point, we had fully validated LYN in vitro. So we wanted to move to a in vivo model, and we generated mice that do not express LYN specifically in their beta cells. So we deleted that LYN gene only in beta cells. And what we found is the following. So invalidation of LYN precipitates diabetes and also prevent beta cell regeneration. So as you can see in the picture below, mice that do not express LYN on the right-hand side have smaller islets, and they show worsened glucose tolerance and then diabetes in later life. So at this point, we had characterized LYN as a critical regulator of beta cell mass. both in beta cells but also in animal models. And that rose the question, are there specific activators of Lin that we could use pharmacologically? So small molecules or drugs that we could use to manipulate Lin in patients, hopefully. So, guys, um, so searching the literature, we found a small molecule activator of Lin that was first discovered and developed by Pfizer as, as a treatment for gastric ulcers. And that molecule is called MLR 1023, as you can see right here. But it was abandoned by Pfizer as a medication for gastric ulcers for lack of efficacy. So it was not really working on gastric ulcers. But then it was years later discovered by Melior. It was discovered that this molecule exerted glucose-lowering properties in type 2 diabetic patients. So this is what they have published here. So you see controlled patients receiving a placebo. So over the course of 28 days, their blood glucose went worse, and that's why the area is shaded in red color in between the two curves. So at the beginning and at the end of the study, versus those who received MLR 1023 for 28 days, you see an improvement in glucose tolerance. So the area in between the two curves is colored in green to show an improvement. So glucose was lower after treatment with MLR 1023. So for those of you who are not familiar, in this essay, people with type 2 diabetes were subjected to a glucose tolerance test. So they drink a sweetened beverage, and then we measure blood glucose over time, over the course here of 3 hours. Ideally, what we would like to see is glucose coming back to normal levels during that time. And then here you can observe the effect of MLR 1023. So the two curves are statistically significant. And it was lower in the case of people treated with MLR 1023. So at this point, we contacted Melior, and then we shared our results. And needless to say that they were pretty enthusiastic. They were excited by our results showing that MLR 1023 was actually targeting beta cells to exert their action. So I asked the question, could MLR 1023 be used to treat type 1 diabetes? And what we've done is treated mice with type 1 diabetes with MLR 1023 for 7 days. We did so by measuring glucose in these mice. And then as soon as we detected diabetes, then we started treating them with either a placebo or MLR 1033 for 7 days. And the results are shown here. You see an improvement in blood glucose in these mice that receive MLR 1023 in red. And also when we subjected these mice to a glucose tolerance test, like I described before, you see an improvement in glucose homeostasis or glucose control in these mice. So you see the red curve is lower than the black curve and becomes significant here at the end. So that showed that MLR 1023 improved glucose tolerance in mice with type 1 diabetes with a treatment period as short as 7 days. And then we measured the effect on beta cell mass. So you see here a nice expansion of pancreatic beta cells with MLR 1023 versus a control islet on the left-hand side. So you see the control islet is smaller. Whoops, sorry. It's smaller. And then after treatment with MLR 1023, there's this nice expansion and protection of pancreatic beta cells. You see that the effect here is almost 2 to 3-fold. So we increase beta cell mass by a 2 to 3-fold factor after only 7 days of treatment. We can do multiple other measurements, such as measuring the infiltration of alpha cells in the middle of the islets. We can measure also the morphology, the architecture, such as islet roundness also is another indicator of beta cell health or islet health. And then all these parameters were all improved after MLR treatment. So you see, for example, in the control islets, lots of dark area. So there's like regions in the islets that do not express insulin or hormones altogether. So you see that this does not happen in islets that were treated or animals that were treated with MLR 1023. So at this point, I could summarize our project by saying that MLR 1023 seems to be a new and promising medication for type 1 diabetes. It promotes beta cell mass expansion, so as much as twofold increase after 1 week. It protects residual beta cell mass, so those surviving beta cells are protected and they do not die as much as in the controls. It improves glucose control as well, and then it delays disease progression, at least in mice. I would like to also say that if I refer to the Melior experiment in people with type 2 diabetes, so we now know that after treatment, if we remove the medication, those patients still display improvement in glucose controls after a few weeks. So a few weeks after drug withdrawal, they still display improvement in glucose controls. And to me, that supports my hypothesis that MLR 1023 acts on pancreatic beta cells and possibly on beta cell regeneration, because this effect is long lasting even after drug withdrawal. So if MLR 1023 was acting acutely to regulate glucose, when we remove the drug, there will be no effect, no remaining effect. So the fact that there's a long lasting effect, to me, supports my hypothesis that it changes something permanently in patients that receive MLR 1023. So at this point, we propose that there's an urgency to test MLR 1023 in participants with type 1 diabetes. So we want to, with my industrial partner, Melior Pharmaceuticals, so we want to launch a small proof of concept clinical trial to test the efficacy of MLR 1023 in participants with type 1 diabetes. If our prediction is correct, those patients that receive MLR 1023 will have their pancreatic beta cells protected from the autoimmune attack and from dying. We would stimulate beta cell mass proliferation and expansion, and that will translate into improved glucose control and reducing insulin use. And if we are fully successful and our expectations are met, people that are treated with MLR 1023 would become insulin independent. I think, so I wanted to show this figure because I think it supports my great optimism and enthusiasm in this project. And it's because here I show the development of the process of drug discovery and development over time. And first, it shows that we start with 10,000 compounds that we have to test and validate in the lab. Out of those, only 250 will be promising, and then will be tested in preclinical models. So that means in mice. Then we'll assess their efficacy and their safety. And of those 250 compounds, only 5 will be successful and be tested in the clinic. And out of those 5 molecules, average, only 1 of them will become a real drug. And in the case of MLR 1023, and that process, I forgot to mention, that process takes almost 20 years. And in the case of MLR 1023, we're already here at this stage where we could perform a phase 2 clinical trial. The reason is, so we have validated those molecules. We have validated those molecules in cell lines, in human islets, and in mice. And now we can also skip those preclinical and safety or toxicology studies, because thousands of patients have received MLR 1023 already, thanks to the studies performed by Melior and also Pfizer when this molecule was considered a medication for gastric ulcers. So we know about the safety, the tolerance, the toxicology, the pharmacokinetics of that drug. And then, so that allows us to fast track MLR 1023 and launch already phase 2 clinical trials, thereby saving us years and years of development and research. I would like to conclude with this figure here that shows one of my favorite articles. So at this point, usually a question I get is, why would you target beta cell regeneration when we know that these cells are under attack and that they are destroyed in type 1 diabetes? So it was once thought that patients with type 1 diabetes had no surviving beta cells, so that the autoimmune reaction destroyed all those beta cells until they were absent. But we now know that it's not true. So there's accumulating evidence, so a huge body of literature showing that patients with type 1 diabetes are insulin microsecretors. So that means that they have functional surviving beta cells. And in the case of this paper, that's even 10 plus years. Here they say 30 years duration with the disease. So with long history of the disease, people still have functional surviving beta cells. So to me, the Saint Grail of diabetes treatment will be to harness those cells that are just slightly different, that they evade the autoimmune destruction. So we could target those cells, stimulate them to regenerate a functional beta cell mass. So to me, that will be the ultimate goal of type 1 diabetes treatment. So I will stop here, conclude my talk here, and maybe answer your questions if you have any. Thank you.
Jean, thank you very much for your presentation once again. Questions from anybody that's participating?
The question I was going to ask you, and you did answer it. You know, we always thought when a person's diabetic that they had no beta cells secretion, but in fact, you're saying that there's proof now to show that you have continuing beta cells, even though if you're, have diabetes for long term. But when you increase the beta cell mass, do you think that comes from the islets themselves, or from the progenitor cells and from the duct cells, or various other cells within the islet?
So in the case specifically of MLR 1023, we show that there's a huge effect on residual beta cells, so those that are surviving. So they proliferate more, they evade destruction, and that contributes to beta cell regeneration for a fact. But of course, we cannot exclude an effect on progenitor cells that will also reside in the pancreas and could give rise to new beta cells. So of course, we did not exclude that specifically. But from all of our experiments, we know that there's at least a huge effect on beta cell survival and beta cell proliferation.
And I think you alluded to, or you mentioned that, in fact, the effect seems to be long lasting. So, for example, you treat, an animal or whatever, and then you stop the drug, the animal is still protected, or eventually will it fail?
Yes. So that's from that's from our own animal studies, but even more powerful from the Melior study in phase 2 clinical trial in type 2 diabetic patients. So those that were treated with with MLR, they had a follow-up visit 2 weeks after treatment, and their blood glucose levels were still, or their glucose tolerance was still improved. So that's even like 2 weeks after drug withdrawal. So to me, that's a very huge indication that there's a permanent effect of that drug. They have higher insulin levels. To me, that's consistent with an effect on beta cell mass, one that we see in animals and in vitro models as well.
Okay.
Hi, John. Excellent talk, and congratulations on your success. Just kind of a follow-up to Dr. Rajat's questions. I assume you looked at like proliferation in generals, like Ki-67 staining or something like that. Was that isolated just to the islets or to the beta cells, so like co-stain with insulin and not like the acinar tissue? And then maybe this is more academic, but if you thought to look at like lineage tracing for if they're de-differentiating, say, alpha cells to beta cells, which there's some evidence at very low beta cell mass that'll happen, or from precursors like the ductal cells.
So your first question was, is proliferation restricted to islet cells? And it looks like it's the case. I don't want to flip slides in front of you, but if you remember, oh no, I didn't show it, but Lin expression, first of all, is restricted to islets in the pancreas. So that limits potential effects of MLR on the acinar tissue. Also, we have measured Ki67 staining, PCNA staining as an index of proliferation, and it's restricted to islet cells. So we know that. So only islet cells will proliferate. Your second question was, could we perform lineage tracing? So we did it. So we did one experiment. We lineage trace neurogenin 3, which is a marker of beta cell differentiation from progenitor cells, and we didn't detect any staining. So there could still be a contribution of alpha cells transdifferentiating to beta cells, because that will not require neurogenin 3. It only excludes the potential contribution of progenitor cells or stem cells that will have to go through the neurogenin 3 pathway to become beta cells. But there could be other possibilities as well. So we can only exclude that one arm among all the multiple scenarios that you can envision. So only that one arm.
Excellent. Thanks, John.
Hi, I have a quick question. This is Tatsuya from Arlet Lab. Is in vitro effect of this compound confirmed, or have you ever done the in vitro experiment?
Yes. So we start with cell lines, but obviously then we go to human islets to be as close as possible to the human physiology. And then we found that MLR 1023 was protecting beta cells from toxic environment, but also stimulating beta cell proliferation. So human beta cell proliferation. So in the control situation, we don't detect any beta cell proliferation in human islets, but when we treat with MLR 1023, for 1 week, we see as much as 2 or 3% of the cells proliferating, depending on the donor.
Jean, the question, as you know, in islet transplantation in the human situation, the problem is, as you know, IBMIR, and there's a lot of cell death immediately around the time of transplantation. So example, if you transplant, the number doesn't matter, 10,000 islets, only maybe 50% survive or less. Do you think using your drug would sort of help that?
So the previous ADF funding that we received, so the small pilot projects, we tested exactly that possibility. So we treated recipient of marginal islet mass with MLR 1023, and we detect a small improvement in glucose control. It's very weak, but we detect better engraftment, increased beta cell mass, and potentially increased vascularization as well after islet transplant. But those results are preliminary, so they're probably not ready to be presented. So we need to accumulate more mice, just because there's lots of variation in between each recipient.
Time for one more question. If not, Jean, I want to thank you once again for taking time this lunch, and for everyone joining us today. Really appreciate it. Very exciting times at the University of Alberta among a very skilled group. And Jean, we wish you all the best with this. I want to leave you with a parting thought that for some participants last week, your presentation gave them great hope and for some that have had diabetes for almost 50 years, was the first hope that they had heard in a very, very long time. So you have a lot of people rooting for you, Jean, and
Hey, thank you, Brad.
we do wish you all well. So
Thank you, Brad.
thank you very much. Thanks everybody for joining today. Really appreciate it.
Thank you all.